Okay, welcome back to part 3 of chapter 41 on animal nutrition. We just got done talking about the whole entire human digestive system and all the parts involved, cells involved, enzymes involved, etc. So now, kind of wrap up the chapter with some adaptation stuff, which is the central theme of the entire Bio 1407 course. So here are some digestive adaptations that animals have evolved over time to the type of food that they eat, which is their diet. So one of those adaptation is dentition. Dentition is the sort of assortment of teeth that animals have that is adapted to what kind of food they ingest. So the type of food they eat will determine the type of teeth they have. So that's what dentition is. So for carnivores, such as dogs and cats, they tend to have pointed incisors like that, pointy front sharp teeth, which are the incisors teeth, and then the canine teeth are also pointed where it helps them tear things and tear whatever flesh they're trying to eat from their prey. And then there's also premolars and molars. So they're kind of color coordinated according to this legend right there. If you're wondering which is the incisors, which are the canines, premolars, etc. So premolars and molars then kind of do the job of crushing and shredding the food before the animal can swallow that piece of meat. For herbivores, on the other hand, such as horses and deers, they don't need sharp teeth to tear very sharp teeth, pointed teeth to tear plants apart. So they still have incisors and canines, and actually some herbivores don't have canine altogether, but they are much flatter in shape, and they're sort of shaped for the purpose of biting off pieces of plants, so which is why they look flatter in the shape of these incisors. They can just kind of slice the plants in half, for example, and then the premolars and molars then will have broad and rigid surfaces to grind that tough plant material into a mush, a bolus, before it will be swallowed by the animal itself. And last but not least, you have omnivores, which is us humans, for example, and we have a whole range of teeth shape for our selection of diet that we can eat. So there's a total of 8 blade-like incisors. They're like little knives that are cutting off and chopping off small little chunks of food as you're putting it into your mouth, and then for biting purposes. And then you have 4 pointed canines for tearing purposes, which is why those are a little sharper, but that's still not as sharp as those carnivores we see, but it's sharper than your incisors. And then 8 premolars are in the back for grinding, 12 molars. We're talking about upper and lower jaw and left and right. So that's why there's 12 molars for then crushing purposes. All right, then other than dentition adaptation, stomach size and also intestinal size also are adaptations to differences in diets. So they have found that stomachs are usually very large and expandable in carnivores, mainly carnivores, or us humans, omnivores case, because we may have to wait a long time between meals before they can find another prey. So that's why usually for carnivores, they will eat as much as they can when they have the prey on hand so that they can last for hours or even days. And in the snake's case, it was weeks before they need to eat again because they may have to wait a long time. Don't know when that next prey will come by in the forest or in the wild. And sometimes you might not be successful catching one as well. So that's why you have very large expandable stomachs. And what allows the stomach to be enlarged are little fold things called rugae that we talked about in lab. So rugae is folds that allows that's why if it's an empty stomach, the inside actually looks kind of wrinkly. I'm trying to draw the stomach here. Okay, so the inside is actually very wrinkly because when it expands, let's say to a big balloon or something, then all those folds then folds out, sort of smooths out and give you the extra space. Now, we already talked about python ingesting a whole gazelle. Lions can actually consume 1/5 of its weight. So if they weigh 200 pounds or kilograms, they can actually ingest 40 kilograms of food and not have to eat for a long while. And it's for hours or a day. And then so alimentary canals are also adapted for whatever diet they're eating. So herbivores and omnivores actually have longer alimentary canals. So they have longer in terms of length That's because plant matter, because both herbivores and omnivores will eat plants. Plant matter takes a lot longer to digest due to their cellulose in the plant cell walls. We already talked about for humans, we can't even digest them at all. Um, but they do go in the way of all the food that we eat. So that's why we have a longer digestive tract or alimentary canal. But think about herbivores in the wild animals that are just eating on plants. So they need to give a lot more time and surface area to maximize that small amount of nutrients they can get from all the leaves, bamboos, plant matter that they eat. So pandas, koala bears, etc., they have a much longer that alimentary canal. This is their small intestine. Look at that whole big cecum they have, along with the colon length. So this is for specifically your koala bears or koalas, which have they're only relying their entire nutrient intake from just tiny little eucalyptus leaves. And so they are very nutrient poor when we look at how much nutrient you can get per leaf. So they just keep eating it all day, letting it, digest through their long alimentary canal tract. Also, they need actually bacteria to help them digest all that plant matter, that cellulose, break the cellulose for making energy that they need to survive. Which leads to our next topic on mutualism. Mutualism is talking about interaction between two species that benefit each other. So it's nice for them to live together in a mutual relationship because these two population of species actually benefit each other in their own little way. So for example, there's a big mutualism between bacteria and our digestive system, whether it be herbivores, omnivores, or carnivores. We have bacteria in the digestive system for enhance of digestion in some way, one way or the other. So, some intestinal bacteria, for example, can produce vitamins for us. They produce vitamin K, biotin, or folic acid. Other intestinal bacteria can regulate development of intestinal epithelium. That's cool. It gives them more ways to turn over, maybe, allows the cells to turn over quicker, and then also function as what we call the innate immune system. Innate means something that we're born with. We don't have to acquire it. As soon as we're born, we have this particular part of our immune system for fighting off bacteria. And in this case, they are our innate immune system because the fact that we have all these good bacteria colonizing our large intestine kind of fend off whatever harmful bacteria that are passing by. So let's say there's all these good bacteria that is filled on your entire large intestine, have taken every spot possible, and then you ingest something that's have harmful bacteria in it. So as that harmful bacteria is going through your large intestine, it actually then have to compete with your good bacteria that's already living there in order for them to colonize. So sometimes they fail and they cannot fight, they cannot compete enough, and those bacteria, bad bacteria, will then just come out of your feces without harming you whatsoever. So this is why it's important to have good bacteria in your in large intestine and your gut in general. And there's bacteria in the stomach too. For proper digestion, which is why we need probiotics and why we need what's called a microbiome in the entire body, which is a collection of all the microorganisms that are living in or on your body. That's what we call microbiome. And I think they say that a microbiome contributes to a certain amount of weight of our body, a pretty good percentage, even though they're just microbiome. Um, and it is a study of microbiome that lead us to know more about diseases and also microbes' role in human health. So this is why people start studying microbiome of human populations or animal populations for that matter. One example is a particular bacteria called Helicobacter pylori, okay, or we also call it short by H. pylori. So Helicobacter pylori is what causes gastric ulcers within the stomach. And it used to be the case where no one thought that any bacteria can live in such an acidic environment as your stomach. They think bacteria should die in such an acidic environment. So And it kind of there's a whole documentary about the guy that discovered it was bacteria that caused stomach ulcers, because people that had stomach ulcers before this discovery thought, oh, it's stress and spicy food and fried foods that's called giving you stomach ulcers. So they just give them antacids and more antacids, and your stomach just keep churning more and more stomach acid, because the source of the stomach ulcers are not the stress or the food that you eat, but the bacteria that's living in there. So it was so, scientists can be so subjective in their beliefs that it took one of the scientists, or the guy that discovered H. pylori, he drank and ate this bacteria himself and showed to the world that, hey, I got ulcers from the bacteria. That's when people are like, oh, well, there's bacteria that can live in stomach acid, maybe. Okay. This really took it to that for scientists back then to believe that bacteria can live in there. All right, so people now treat, H. pylori infections and stomach ulcers with antibiotics that can kill the bacteria itself. So microbiome has been analyzed to learn why H. pylori infection leads to ulcers. Well, it turns out that when we look at the entire microbiome of your stomach, when you have people that is infected with H. pylori, which is represented by the green bar there, all of the other bacteria species that's supposed to be in a healthy gut system is gone. Okay, so which means H. pylori just have a very good competition capacity, that they can outcompete all the other bacteria that's supposed to live in your gut nicely, all the probiotics and stomach bacteria that's living there normally. They outcompete it. And now all you got in the stomach is just H. pylori and nothing else, and which then leads to stomach ulcers. Whereas those that we see as healthy individuals, which is represented by that purple bars there, have a nice variety of bacteria that's living in their stomach, working as their microbiome probiotics. So, study of microbiome has also led to new approaches to treatment without antibiotics. It's sound kind of gross, but it's pretty good, because the research on trying to come up with new ways to fight bacteria without antibiotics is always a cutting edge research. Is what we human race is, or public health is trying to do. Is because there's too many strains of different antibiotic resistant bacteria, so we cannot rely on antibiotics forever. As we have described in the previous, chapter in the beginning of the semester, when we have introduced new antibiotic to a bacterium, they would just create new adaptations, or they would be selected for their new adaptation to that new antibiotic. So we constantly have to play a catching game with them. But if we have another way of killing them without antibiotics, that will be nice. It serves as another approach. So this particular case scenarios, there was this bacteria called Clostridium difficile. Okay, Clostridium difficile. And I'm not very good with those scientific names. So Clostridium difficile then is the bacteria that have caused some form of untreatable diarrhea for the longest time, until we have tried a new method to help people with this particular bacteria. They found that usually this particular bacterium infection is only common in individuals that have, let's say, taken a round of antibiotics. Antibiotics have, not only does it kill what you don't want in the bad bacteria in your body, it also kills off the good bacteria that's supposed to be living in your gut. So when you're taking them all out, Clostridium difficile will then take that chance and start colonizing your whole intestine or digestive tract, which is why they can thrive in after all the intestinal microbiome is gone. So they have found one way to treat or kill them without antibiotics, or prevent them is kill them, is by fecal microbial transplantation. So it's really taking microbiome from a healthy individual and introduce it to the patient's intestines. You're like, ooh, you're getting someone else's poop, but hey, it's the microbiome that you're getting from them. Okay. And they have found this to be an effective treatment against Clostridium difficile. If it wasn't for fecal microbial transplantation, this diarrhea, this particular bacteria, will remain untreated forever to come. But because of this new method, it holds something promising. If our natural microbiome can kill something for us, that's even better. That we don't have to rely on new antibiotic approaches. All right. And then continuing on with mutualism is that some microorganisms help with herbivores to adapt to their plant diet, as I've talked about earlier. I already said that animals, humans, well, herbivores or not, they lack enzymes to help them digest the plant's cellulose for energy. For humans, our bacteria can't even use cellulose for energy. So as an adaptation, herbivores host a large population of mutualistic bacteria or protists for that matter, which those guys then have the enzymes to digest cellulose for them and release simple sugars and fatty acids for that animal to survive and get their nutrients from. So that's why you need those fermentation chambers we talked about in terms of their alimentary canals. Sometimes there's multiple compartments, which where their stomach is divided into multiple compartments, or their cecum is longer that will allow for all that fermentation to happen. Like I've said, that cecum is for that purpose of giving them a bigger compartment for that symbiotic bacteria, protist to do the job for them of digesting cellulose into their individual monomers for energy. So horses, koala, elephants have symbiotic microorganism in their cecum. Hoatzin, which is a bird, actually hosts microorganism in their crop itself. Everybody, animals have, their own adaptation of where they host these or organisms for digesting cellulose. Excuse me, again, not rabbits. Rabbits, rodents, have symbiotic bacteria in their large intestine, including the cecum. So because of the fact that they, kind of like humans, only have nutrient absorption within the small intestine. So once food, chyme, all that stuff has passed through small intestines, nutrient absorption is done. Okay, done deal. So what happens is that cellulose that's being digested in the large intestine now goes to waste if they don't ingest that back, which leads to the idea of coprophagy. Okay, coprophagy is when you are eating feces, feeding on feces to pass through the entire alimentary canal a second time around. So you will see some rodents that will eat their own feces. And again, that's all for their good purpose of allowing that nutrient, which is broken down from cellulose by the bacteria that live in the large intestine, and give this now the broken cellulose a chance for small intestine to absorb. So that's why there is that, eating species type of adaptation for these particular animals, so they don't lose those, very critical nutrient that they can get from cellulose, especially if they're herbivores that only rely on plants for energy and nothing else. Now, ruminants are actually cud chewing animals. Cud meaning that these are regurgitated material. So this is usually prominent in deer, sheep, and cattle. We call them ruminants because they actually regurgitate a lot of their food so that it goes through different compartments of their stomach or the whole alimentary canal. So they have actually evolved what they call the most elaborate adaptation for herbivorous diets. So usually what happens is, imagine, let's say cow, for example, right here. So cow chews their grass. The grass first enters through the mouth, goes into the esophagus, will go into the rumen and the reticulum. So some of it will go into the rumen, which is all this compartment right here, and some of it will go into the reticulum, which is right here, this pouch, and they kind of get churned and digested throughout those two compartments. And then some cows will actually regurgitate that material so that things from the rumen and things from the reticulum will come back out the esophagus, go into the cow's mouth again, and the mouth starts to, the cow starts chewing, which is why you see cows chewing when they pretty much all the time. They're just constantly chewing and chewing and chewing, even though you don't see them grabbing grass. It's because they might be chewing some of that regurgitated material and then swallow it all over again down their esophagus into another compartment of their small of their oh discard. Excuse me, let me go back. All right. So once the regurgitated material is re-chewed a second time, go through the esophagus into another compartment called the omasum, which is a third chamber of their gastrointestinal tract, and then pass through to the obomasum. Okay, obomasum, which is then the fourth chamber for the rest of that food to go into the cow's intestine for absorption of those important plant matter materials that they can break down. So it is for cows, they have these different compartments inside their stomach that can do all this stuff. So that's a little different with, the horses, hoatzins, or the rabbits, where most of them are in either in their crop or the cecum or large intestine. For ruminants, they have those beneficial bacteria inside their stomach in different compartments instead, and let that grass and plant material go through multiple times inside the stomach without having to eat their own feces, because they can make sure things are digested before it goes into their intestine. All right, and then the last part of this chapter, just a big summary thing. I don't want to go into too detail about hormonal regulation, but it is a part of digestive process, a very important digestive part about digestion is the hormone that tells your stomach when to start churning, tells your intestine when to secrete those particular enzymes, because you don't, like I said, you don't want to be digesting when there's no food in that particular compartment, or else you will run into a stomach acid issue. There's no food to digest, but your stomach is just releasing stomach acid into the environment, wasting it all away and burning those mucus because there's no other food in there to digest. So there's hormones that regulate when the stomach, saliva, all that stuff release what it secretes whatever they need to. So the endocrine system then comes into play with hormones, because it is the endocrine system that releases hormone. Nervous system releases neurotransmitters. And so for endocrine system, they communicate through hormones, and hormones goes into the bloodstream. So it's a much slower response when we're thinking about in terms of nervous system response. Nervous system response is more of a quick turnaround, within milli of a second, tenth of a second, you can have a reaction. Endocrine system goes through the bloodstream, slowly cause something to happen. So a couple of those hormones that is in charge of digestion is one of those is called gastrin. Gastrin is a hormone that is released by the stomach, and when food arrives in the stomach. So once food goes into the stomach, it stretches the stomach. So when the stomach senses that there's baroreceptors in there that tells it, hey, there's something coming into the stomach, this is what triggers the stomach cells to release gastrin. And gastrin is then what tells all the other cells, parietal cells, chief cells, and to start, really secreting those gastric juices, hydrochloric acid, pepsin, into the lumen of the stomach, and starts churning away and mixing that bolus into chyme. Next hormones, so cholecystokinin, which is CCK, is released by the duodenum. So this is the duodenum right here. So we're talking about chyme having moved now into the small intestine. Now it is CCK then tells the duodenum um, to stimulate the release of digestive enzymes from the pancreas and the bile. Like I said, you don't want them to be really wasting all those good enzymes away unless food or chyme has arrived at the duodenum. So it is CCK that tells the pancreas and the liver, hey, it's time to do your stuff, or gallbladder to do your stuff and release those important digestive enzymes. And don't forget secretin. So secretin and CCK works together. Secretin is then what tells, again, when chyme arrived at the duodenum, duodenal cells will secrete secretin. Secretin goes to your pancreas, tell it, hey, acid has arrived. Come release bicarbonates. Pancreas will release bicarbonate as a buffer to neutralize the chyme so that the chyme's acid will not burn your intestinal tissues. And then you have two other hormones that has to do with your satiety and hunger feelings, more of a behavior type of thing. So ghrelin turns out to be a hormone that is released by your stomach wall when it's empty, that tells it, hey, my stomach is empty. You got to go eat. So this is what stimulates the hunger feelings before meals, because your stomach is empty. So ghrelin gets released, telling your brain, hey, you got to go eat, and you look for four sources of food. Whereas after you eat, you're full, your stomach is full, intestine is full, then it releases leptin. Leptin is released by actually fat tissues in your body, and that's actually suppresses appetite after you finish a meal so that you know when you're full. No one will just keep eating and eating and eating until they vomit, unless they have no control whatsoever by leptin. Um, in addition to leptin that's released by adipose tissue, there's also insulin that's released by the pancreas and PYY, another hormone that have the same effect of suppressing your appetite. So multiple things that make sure you don't overeat and eat until you vomit. And the last two hormones we'll talk about has to do with blood glucose homeostasis. Remember, homeostasis is the balance. And whenever we eat a lot of carbohydrates, all of them get broken down into their individual monosaccharides, such as glucose. So all that glucose then from your intestinal epithelial cells gets absorbed into your bloodstream throughout your the entire circulatory system. So once sugar appears in your blood circulatory system, there's only a certain amount that it can keep circulating around, which is about 70 to 110 milligrams per 100 mils of blood. So once the level of blood goes above, a level of sugar goes above that range in your blood, there's a signal that it tells your pancreas, specifically beta cells in the pancreas, to release a hormone called insulin. So pancreas has a lot of jobs too, kind of like, well, liver even more, but pancreas has, buffering capacity, digestive enzyme capacity, and endocrine hormone producing capacity for glucose homeostasis. That's pretty cool of pancreas. So pancreas beta cells release insulin. Insulin is a hormone that tells your cells to absorb glucose. It's kind of like, hey, everyone, there's a lot of glucose in the bloodstream. We got to start absorbing them, or else it will have detrimental effects in the entire body. So that's why then insulin tells your cells to uptake all those extra glucose from the bloodstream, and especially in the liver, and then we can store them as what we call glycogen. So, having a lot of glucose in the blood is a bad thing, but having a lot of glucose in the liver just turns them into glycogen, which is our bank, our big bank of glucose, so that we can use it when we need glucose again. So, usually, insulin is released after a carbohydrate rich meal, and the only exception where a cell would just absorb glucose regardless is there or not is brain cells or neurons, because they constantly need ATP. So regardless, we're sleeping, resting, running, doing whatever, in order for us to live, our neurons will constantly use and metabolize ATP. So which is why they need a constant supply of glucose. They don't need insulin to tell them what to do because they need it all the time. There's too many things going on that need ATP, such as electro signal conduction. If we don't have electric signal conduction, we cannot move, for example, for our skeletal muscles. So this is why neurons need glucose constantly, regardless what we do. And there's always also the priority for glucose delivery. Even if we're starving, we try your body will always deliver glucose to your neurons. All right, because or else you will die once your neurons shut down. Okay, so then the other way around is then if, let's say, you ate the meal and then you didn't eat for the next 5 hours, 6 hours or so, everything has been digested from your stomach. Now you have empty stomach. So between meals, glucagon is usually released, which is a hormone released by the pancreas's alpha cells. Um, so good way to remember when glucose is released. Remember glucagon, glucose are gone. Okay, so glucagon, glucose are gone. No glucose, not enough glucose is in the bloodstream. Now you need to release glucose back into the bloodstream. So that glucagon will go to your liver cells, tell it, hey, we're not having enough glucose. Break down those glycogen, release those glucose into the bloodstream so that our cells can use it to keep them alive. It also goes to adipose tissue, of course, other than liver. Let's say if there's not no more glycogen left, and then their next source is the fatty acids for, supplying their body with energy. So usually glucagon are released between meals. This whole idea of these two antagonistic relationship of hormones to regulate blood glucose homeostasis is also shown in this particular picture here. I won't go through each of them in detail because I already went through it by words. But if you're in a pictorial form, that's a pictorial form of blood glucose homeostasis regulated by your pancreatic cells, beta cells and alpha cells. All right, and that I just want to end the chapter with a quick thing with what I did when I was an undergrad. I actually worked with leptin receptors, and I actually worked with insulin and glucagon studies because I worked at an undergraduate research institute or a real research institute that studies diabetes. So one thing about insulin and glucagon, especially insulin, is that it can lead to type 1 or type 2 diabetes. Type 1 diabetes is when it's an autoimmune problem that such that your beta cells just don't produce insulin or they just start dying. So type 1 diabetes is usually starts when you're young, whereas type 2 diabetes is more of an environmental and genetic factor in that causes the disease. And so this particular mouse is a mouse called DBDB mouse, and the chapter actually talks about an experimental research on DBDB mouse. DBDB mouse is a particular mutation in their genome such that their leptin receptor no longer works. So this is how the mouse is able to become a type 2 diabetes mouse model because we know it has environmental means where people overeat when they that's what or overeat on sugars, fats, etc., that cause them to have type 2 diabetes. So how we mimic that is we take out or block or inhibit the leptin receptor in these mice so that they don't know when they're full. So all day long all they do is just eat. We also feed them a high fat diet. Again, all of this is going according to animal regulations. We're not torturing these animals. Every researcher works with animals have to go through a very strict animal policy and also approval by the animal regulations. So in this case, these type 2 diabetes mouse models serve as a very good way to study what can reverse the effects of type 2 diabetes, because that is always a very common problem in the United States and globally as well, even in Asians, because Asians love rice and we eat too much rice, such that those carbohydrates all turn to sugars, fats, etc., and it elevates our blood sugar levels such that over time, it leads to what's called insulin resistance. So that's what ultimately what causes type 2 diabetes, or the foundation of type 2 diabetes is insulin resistance, where the receptors on our cells just no longer sees insulin as efficiently as they should, and all the glucose just remains in your bloodstream. So this is how we study them, and what can reverse them is by having a type 2 diabetic mouse and also a normal mouse. And you can see there are differences in body size. This one is about usually around 20 grams. These diabetic mouse model is actually 50 grams or more. And they're able to sit on my palm like that because most mouse don't do that. They will just run away right away when you try to pick them up. These don't, because of their body size and their overall life and health levels due to being type 2 diabetic. So when I saw the chapter have DBDB mouse, they talked about that. I was like, hey, I worked with DBDB mouse back when I was in undergraduate research. So this is just, a quick little side story about how I got into biology and fell in love with biology was my undergraduate research experience when I was in college. All right, and that's all for chapter 41. We'll go through the practice question and be done with it. All right, so there's quite a bit of practice questions here because a lot of them were actually good, and we went through a lot of informative things and terminologies, so bear with me. Okay, essential amino acids are what? Those that cannot be made in the body. That's why it's called essential. The fact that it's called essential means you cannot make it yourself, have to be ingested. A complete digestive tract or alimentary canal differs from a gastrovascular cavity in that the complete tract does what? Has specialized compartments. So only complete tract will have specialized compartments like the crop, the gizzard, the stomach, intestine, etc. Whereas the gastrovascular cavity just big one giant hole is really what it is. In humans, the first opportunity for ingested food to undergo enzymatic hydrolysis is I talked about this and emphasized this, the mouth, where saliva releases amylase, an enzyme that starts digesting carbohydrates. Now, we don't start digesting lipids or proteins within the mouth, but we start the carbohydrate process, which is still an enzymatic hydrolysis. All right, hydrochloric acid in the stomach does what? Activates pepsinogen into pepsin, which all the other choices means that is false. Secretin modulates digestion by, and we just got done talking about this, it triggers buffer release from the pancreas. So remember, you secrete, you release secretin for pancreas to secrete a buffer. in that sense. Easy to remember. Okay, bile cell function in fat digestion by doing what? Remember, is not direct digestion of lipids, but dispersing or emulsifying that big droplet of fat into smaller droplets. A fatty acid absorbed into an intestinal cell does what? Or is what? It becomes part of a chylomicron because they're too large to be diffused into into the bloodstream. Mutualistic bacteria aid digestion in many herbivores, including those where this function resides primarily in the cecum. What is an example of that? Horse. All the other ones are ruminants, so they don't need those mutualistic bacteria to be living in their large intestine. It's mainly in their stomach compartments. Glucagon, a pancreatic hormone, functions to do what? Glucose are gone, so stimulate liver to release glucose because glucose are gone. Increased appetite is a typical result of increased levels in the blood of what? Ghrelin. Okay, so when we have ghrelin, remember I do this joke in anatomy. Grr, I'm hungry. Ghrelin. Okay, so you remember ghrelin for I'm hungry, which means increased appetite. All right, increased glucose levels in your blood after a meal trigger what? Release of insulin because now you have a lot of glucose in your bloodstream. You need to take it away with insulin. 95% of the bacterial DNA sequences obtained from stomachs of individuals infected with Bacterium H. pylori are found to be H. pylori mainly, which means all the other bacteria are gone. So the conclusion may be drawn from this finding is that H. pylori outcompetes all the resident bacteria species and kill them all off so that it is the only dominant bacteria that live in the stomach when you're infected with H. pylori. I kind of discussed that when I was talking through the H. pylori story. This is just a reminder of where, that graph I talked about and how H. pylori might look like under the SEM. The primary reason the hepatic portal vein delivers blood-containing products of digestion directly to the liver in order to do what? What does our liver do for us? Detoxify foreign molecules and metabolic wastes. A simple change contributed to the improvement of human health by reducing the risk of neurotube defects. This change was what? Addition of folic acid to grain products, the very famous, very effective program as well. Which of the following cell types is incorrectly matched with this function? Incorrectly. Parietal. So parietal cells do not pump sodium ions into the stomach lumen. If anything, it pumps hydrogen ions and chloride ions into the stomach lumen to make hydrochloric acid. So which of these statements about the role of human microbiome in health and disease is not correct? False. Human mutualistic colon bacteria contain hydrolytic enzymes for digesting cellulose to simple sugars. We wish that's the case. This is why humans cannot use plants for calorie intake because of the fact that we don't have those mutualistic colon bacteria, or else we don't need to ingest other things for sugars for our energy. Which of the following organs or structures is not part of large intestine? Jejunum. Jejunum is part of the small intestine. And that's all I have for you for chapter 41. Know that usually, again, just a reminder, our lectures in class, in person, are usually 2 hours, 40 minutes long. So that's why there are some times where there's long lectures. And this is in short semesters, we have to finish the whole chapter in one day. So, all right, see you later.